Well, welcome everyone. Thank you for taking a minute out of your day and joining us. I know sometimes we can get Zoom fatigue, webinar fatigue, but so we appreciate you registering and jumping on. And those of you, if you know somebody want to jump on, they got tied up, they couldn't. We will be sending out an email with a link to our Redox YouTube page that this will be recorded. Sam from the office is helping us with that, the logistics. She's recording that, and it'll be posted later. Within a couple of days, we'll post that, and you can catch that out. Or if there's a part you want to rewatch, something like that, it'll be there on the YouTube page. Well, happy Thursday, right? We made it to Thursday, and this is another webinar program that we've had in our nut crop series. We started earlier this year, just really when COVID started hitting and some of our meetings had to be canceled. We shifted to doing more webinars, and at various times we'll have guests join us. And today we're really pleased to have Roger Duncan. He is a farm advisor in Stanislaus County, actually director of the UC Cooperative Extension in Stanislaus County. He conducts research and educational programs for growers, pest control advisors, consultants, industry shareholders, and is very knowledgeable. Roger, correct me, I'm probably majority of your time spent on almonds, peaches, maybe wine grapes. Any other crops I'm missing?
No, that's mostly it. We have a few pistachios that are trying to move into our area, so trying to learn about that. So by the way, little shameless plug here, there is a pistachio short course that we're putting on in the first week in November, I think it is. So
first week in November.
Yeah.
Where's that?
And actually it's gonna, again, it's
Virtual?
the Zoom thing, and it's actually gonna be broken up over a few days. So it's like, I think 2 hour, 2, 3, no, it's half days on about 4 or 5 different days or something like that, I think.
Yeah. Okay, we'll look for that. I saw there was an email communication just the other day on the state pistachio day. We usually hold it in Visalia at the convention center, and they just announced that's going to be virtual. I don't remember what month. I don't know. Maybe you'd remember, but I know that was announced too, shifted virtual. So short course, first week in November, to look for that. Okay. Very good. We will have CCA hours available. We are getting that. We submitted. We're waiting for their approval. We should get that. Might even be able to send that out with that follow-up email with the recording. So if you are a CCA and look for hours, look for that. You can reach out directly to us once we get that approval, or you might just see that in the follow-up email, so you can get an hour of your CCA CE units that you'll need. Roger, I don't know if it's appropriate for me to ask, but do you have a favorite crop, that you consult on? Any first loves that you have?
No, I guess almonds, I suppose I would say, but
yeah.
I like peaches. I like grapes.
You like them all. There's little things you like about all of them.
Yeah.
I grew up.
So I'm just really glad. As long as there's shade. I just don't like working in tomatoes and that kind of thing.
Well, grapes, unless it's an open gable, sometimes that gets hot out there.
Yeah,
as well.
Yeah. So that would be on my least favorite.
Well, I do have a love for peaches, cling peaches. I grew up in Northern California with grandparents and uncles that were involved in cling peaches for a long time. So I grew up in a lot of that. So peaches, whenever I drive by or get into a peach orchard, it's always good. It reminds me of being a little kid growing up. So I thought I'd ask. We've got Justin Rose here as well. Justin is an agronomist for Redox as well, and he'll be participating in the conference. And I didn't introduce myself. My name is Jared Sannar. I'm an agronomist as well here in the Fresno area, and appreciate you taking time to be with us. So Roger, you're going to lead us off first. So I'm going to pass the baton to you, and we've invited Roger to share some interesting work that caught my eye. Give you a little background. It caught my eye about compost and orchard performance, and several studies that were done. And so I invited him to share that with us. The soil health, soil biology, it is really gaining momentum. What can we do? What are the budgets we should allocate, and how do we measure these kind of things? So when I saw this, I thought, this is interesting in this arena of soil amendments and what that's going on. So I thought invite Roger, and he was gracious to join us and share his work with us. So Roger, if you want to put your presentation up, we'll
Sure. It says it says I can't share the screen while someone else is participating or is sharing.
I'll drop that. How about now?
That looks better. All right. Okay, so I guess, I mean, I always prefer sort of this a discussion back and forth. So I don't know. I guess maybe I don't know if we're going to do that. I mean, it's Jared, it's up to you. But or if you want to just, I guess, type questions in the chat box. So
yeah, you know what? Yeah, and thank you for reminding me of that. So on this webinar, definitely use that chat feature. I should have had that. There's a Q&A feature on those of you participating, whether in your truck or you're at your office on your computer. There is a way for you to send questions and hit that chat box at the bottom, and we'll catch those and answer them as they come. And I'll try to also, as questions come, Justin and I, we can kind of hit you some questions and filter through that while you're presenting, Roger.
Okay, and you know, again, I don't mind if you just want to unmute and ask a question too. If that, I don't think there's 100 people or anything, so that should be okay. So I don't profess. I'll just start. I don't profess to be an expert in, I guess, soil health. I'm really more interested in is what we're doing. Can you see an effect that the grower can actually, you know, put money in his pocket? So are we seeing an effect on tree response, on yield, or something that saves us money or something like that? So so most of these trials actually have been in response to questions that growers are asking me. You know, does it make sense to do this sort of a thing? So there we go. All right, so we know, you know, putting compost on increases a lot of measures of soil health. So we know that it can improve organic matter, it can increase the cation exchange capacity, microbial activity, and that kind of a thing. But the real question, again, to me, is it produce a meaningful change that a grower can benefit from economically? So I'm just going to quickly go through a few different things. So this first one was we looked at pre-plant and post-plant organic materials and microbial materials and things. This is in a peach orchard. So this is an orchard that is very sandy ground. It was replanted. I think this is probably at least the third generation orchard. And so this was done quite a while ago. Matter of fact, this photo was an actual photo of me hanging out an airplane taking a picture. So this was prior to using Google Earth. So but you can see in this sort, so this is a looking at pre-plant combined with post-plant materials afterwards. So you can see that we fumigated, in this case with methyl bromide, going across the rows. And then here is the check, and then here we use BAPAM and repeated it again. So there are 1, 2, 3, 4 replications of those. And then we put on post-plant treatments afterwards going down the row. So we would have sort of this checkerboard. So we have materials going on with and without fumigation, if that makes sense.
And Roger, can I ask, was that peach following peach?
It was peach following peach, probably following peach.
Okay.
Lovell, actually. Lovell. So again, we look at different microbial materials as well, but the main thing was putting on compost, and then on top of the compost, we would add sometimes other materials. So sometimes the trees were dipped, and then we put in microbial amendments injected through the drip system monthly. And there's the compost. So the bottom line there, what we found was that where the compost went on, I'm going to put this down. Where we put the compost on, we increased the microbial activity, but only where the compost went on. So where we put compost on, when you add carbon to the soil, you know, anytime you put a whole bunch of food in the soil, anything that feeds on it is going to increase in population. And so that's what we saw. We saw that where we put the compost down, we saw a corresponding increase in microbial activity. We did not see an overall effect using these microbial inoculants that we were looking at the time. And then again, looking at just different things the grower might care about. So here we have a photograph. I think you might be able to see my cursor, I hope, but down the bottom right, we see the prunings from where we had where we fumigated, and then this is not fumigated. So massive difference in tree growth. So in this case, we're looking at the pruning weights. We have unfumigated methyl bromide and VAPAM, and then going this direction, looking at compost, and then with microbial materials and things like that, humic acid and such. But the bottom line was that we saw here was a massive amount of, we saw a real effect where we used methyl bromide and some with VAPAM, but not really any effect at all on tree growth. You're looking at, using the compost, or even when we added microbial materials or microbes and kelp and humic acid. And then this just looks looking at the yield, kind of the same story. No, we saw, big yield effect where we fumigated versus not fumigated. Not really an effect that we could measure with the compost or the microbial materials that we injected. So bottom line, after 3 years, we had no real effect on anything that we could measure really using compost. So this is after, was it 2 or 3 years of these treatments. So no effect on yield, size, tree growth, tree nutrition, nematodes, anything that we could really measure. We didn't see any effect. So now fast forward to, 2015, I think. So this is in response to a nursery who was looking at, they wanted some information on their customers who were buying trees. The nursery wanted to give recommendations with how much compost to put out. And they were going to look at like 1 ton, 2 tons, 5 tons, that kind of a thing. And I told them, based on my past experience, I think we really need to increase that. So we looked at 5 tons, 10 tons, 20 tons, and 30 tons pre-plant. And this is really pretty good ground. Had a vineyard prior to this, and then trees were going back after that. And so this was the application. So you can see that there's a little bit of a kind of a ditch that was built with the kind of the pre-plant thing that they use to break up the soil, to rip the soil, and it left a little bit of a depression. So what you're seeing right there is about 10 tons of compost per acre, all concentrated into one band. And so here this is 5 tons, 10 tons, and then this is what 20 and 30 tons look like. This is also what those cost. A fairly significant investment.
Come back and fold that in when they made the berms?
Pardon me?
Did they fold that in when they made the berm before plant, or they just plant directly into that like that?
Yes, they use a machine. I don't have a picture. I didn't have a photo with that, but it was all folded into the, it was all folded into the berm at the planting time. So here's so I guess what I wanted to show here was that, so this is the amount of NP and K that went on with those applications. So, it's about 1.25% nitrogen as delivered. So that's roughly 25 pounds of nitrogen per ton. Now, compost really is variable, but roughly, it's roughly 40 to 50% water. But as delivered, it's about 1.25%. So roughly 25 pounds of nitrogen per ton. So if you're putting on 10 tons, you're looking at 250 pounds of nitrogen, which is going to be a red flag potentially, I guess, when you do your nitrogen use reports. And there is really a lot of disagreement about how much of this nitrogen is available the first year. If you look in the literature, anywhere between 5 and 20% of that will be available. So it's kind of hard to pin that down, but there is some potassium, definitely. Well, I'm just gonna say we had some treatments where we tried to put on just the nitrogen just to match what we thought was going to be the effect of compost. So in case we saw an effect, we wanted to determine whether it was just due to the extra nitrogen or not. So anyway, so here's the berm that it was incorporated into the berm when the trees were planted and drip irrigated right in. These are potted trees planted in May. So this is looking at, so the question was, does compost reduce water stress of new trees? And so here we're looking at 2 different treatments. The blue diamonds is the check, and then the red square is where we have 10 tons of compost. So this is using the pressure bomb. If you're familiar with that, it measures the amount of water stress by taking a leaf, putting in a chamber here, and then determining how much air pressure you need to put water back through the leaf. And anyway, so the bottom line here is that the more you move up in the scale, the more negative the reading is, then that is a measurement of stress. So so as you move up the y-axis, then it would indicate a little bit more stress. So these are all these measurement dates. For the most part, there wasn't much difference, but you know, fairly consistently, what we see is that the red square is a little bit higher, so meaning a slightly more water stress than the blue diamond. Very, and it bounces around a little bit, but most of the time, you know, we had just that sort of 1/2 of a bar or 1 bar where the compost trees, the trees that were planted in compost, actually were slightly more water stressed. Probably not really significant, but it really goes against what people are saying about, well, if you add compost, it's going to really help with water retention, and you don't have to apply as much water and that kind of a thing. That's just not at all what we had seen.
We've got a question, Roger, if I can interrupt
Yep.
you. We have a question from Tracy. He says, the question is, are the compost rates that were applied in the band down the tree rows, were they on a per planted acre or a per treated acre? Could you give some clarification there?
It was based on a per acre basis. So, but it was all concentrated in that small band.
Okay.
So if you, for instance, 10 tons per acre, if you would have broadcast it, it would have been 10 tons applied to the whole acre, but we compressed that and concentrated that in the band.
Got it.
If that makes sense.
And there's another comment. So another comment from Greg here, question for you. Could that stress be due to the sodium in the compost? It's a question.
It's possible. And actually what we've seen is higher chloride that we're putting the compost out. So that's what I'm wondering, is if maybe it's just a higher amount of salts that we're seeing in the compost.
Okay.
All right, so here, just looking at the trunk circumference over the first leaf and the second leaf, here are all the treatments: 5 tons, 10 tons, 20 tons, and then these are the nitrogen guesses that I was putting out. And essentially, we saw no difference in tree size this first leaf or at the end of the second leaf. So we saw, you know, nothing that would indicate that there was any positive effect from putting these composts on. And we have leaf analyses, and we have some other things too. But the bottom line was that we saw nothing that we could measure that indicated that compost was beneficial. Really, so no measurable effect on tree establishment. So that nursery has decided that they're not going to recommend putting compost out with their potted trees. All right, so then finally, what about if you get on a compost program? So not just a pre-plant deal, but if you are going to sort of put some on pre-plant and then continue on every year. So that's what we did. We've done it for 5 years now in 2 different locations. One is a really nice Hanford sandy loam, virgin soil, you know, just you know, a really nice spot to grow trees with no replant problem or anything like that. No nematodes, no diseases, nothing we could see. And the other site was in North Modesto, pretty sandy ground, and this was a replant site. And this is Independence on Memagard. So here in this case, we'll see how the compost was incorporated into the berm at planting time. So in this case, we did 5 tons, or we were shooting for 5 tons of compost. You can see the berm's made, the tree stuck right into that. And then we came back and we did this little deal here. We put on about 2 shovelfuls of compost around each tree. You know, that actually adds up to just about 1/2 of a ton per acre. So this actually adds up to more than you think. But and then we follow that up with these post-plant applications. So what you see right here is a 5 tons per acre application, and then we turn around, we come back coming the other direction. So it's a total of 10 tons. And again, the 10 tons is concentrated in that roughly 8 or 10 wide band there. So this is the spot that's pretty sandy. This is Independence on Nemegard. So we looked at all these different things, all these different, and then so this cart that I have right here has scales built into it. So when we pick it up with a pickup machine, goes into my cart, and then you can see we have a little computer back here. So there are load cells in the tongue and in the axles, so we can get a really good measurement of yield. And then we can also sample them and it falls down in these buckets, and we take these bags, so we can go back to the office and crack things out and get a pretty good determination on fruit size and that kind, or nut kernel size and all that. So anyway, so what we're saying, so if we look at carbon and nitrogen, and so what basically what we're seeing is that in the top layers of soil, after a couple of years, we do see an increase in soil carbon. So here's the sandy loam orchard, here's the loamy sand orchard, looking at carbon. And so we do see, so this is the untreated, this, if you go to 2018, and then this is where the manure, and then this is the composted green waste. So we are seeing in both locations a tendency to increase carbon in the soil. We're also tending to see more nitrogen building up in those top layers of soil. Cation exchange capacity, fairly similar. So more cation exchange capacity going on in those upper layers of soil where we're putting on, especially the green waste compost. So again, we looked at pressure bombing in these orchards, and we had the same story. So in this case, we'll look at just look at the let's say the check, which is the triangle, the green waste, which is the square. And again, you know, the square tended to always be above the check. So it always tended to be like maybe 1 bar more water stressed all through the year. And it's consistent. Okay, that's hard to see. Let's just skip through that a little bit. So that was looking at trunk circumference. We're not really measuring a lot. We don't really see a lot of difference. So here's a case. Here's a photo of the line is right here. So between a compost treatment and an unamended treatment. So after 4 years, we're putting on 10 tons of compost. I don't think you can see a difference where we've been putting on compost and where we haven't. So I don't know, you probably can't tell which one's which, but going this direction is the compost, and coming this direction is no compost. So we just really haven't seen any difference and haven't measured it yet either. So look, if we're looking at nematodes, in this case, in that sandy spot replant site, we are a little bit concerned with the ring nematode, but here's the check, here's the green waste, here's the manure. This means there's statistically no significant difference. So we're not seeing any difference, and certainly no positive difference on nematode populations where we're putting out the compost. So leaf nutrients, we have the check, here's the green waste and the manure and the urea. So what we've seen is there is a slight increase in nitrogen. Although, you know, if you look at this, you know, 2.6 versus 2.7 or 2.8, statistically it's a different, but agronomically, probably not too much. There is a difference. There is definitely more potassium here where we put the manure on. And we've seen that in other places too, that green waste even does tend to be a good source of potassium over time.
Roger, can I ask you a question? What do you make of the nitrogen, the check versus the green waste manure or just urea treatments with the amount, the tons of compost you're putting out? How many pounds would you say? It was 25 pounds per ton, I think is what you had originally. What do you make of that you're not seeing that necessarily in the tissue?
Well, yeah, so if you're putting on 10 tons, that's 250 pounds of nitrogen, and that's in addition to the grower's normal practices. So he's probably putting on, I have it, but it's got to be, close to 200 pounds. So this was probably getting close to 400 pounds of nitrogen. But I think that the thing is that it's in organic form. It's not available to the tree. And like I said, there's a lot of disagreement as to how much of that nitrogen becomes available over time. Maybe 10%, becomes available each year. So, but then it should be cumulative. You should be getting 10% from what you put on this year, plus 10% from what you put on last year. But we just haven't seen a huge difference. And again, I think it's just because it's in an organic form that isn't available, at least immediately.
I'm looking at the calcium there.
Yeah, that was my next point. And this is very consistent in every place we've done this. And I don't know, I mean, calcium is kind of a, popular thing. But we consistently see lower calcium where we've been putting the compost on. And I'm assuming it's because it may be a sort of a potassium, a reflection of having other cations available in abundance. And so maybe the trees are sort of preferentially taking up, say, potassium, for instance, and not as much calcium. That's maybe. The other thing, again, is the chloride. So we're seeing some pretty high levels of chloride. We're supposed to be staying below 0.3%. So even the check is already over that. So we're now at about double the amount of chloride that we should be seeing in these leaves. Again, not sure that that's a problem, but certainly over time, I think that would show that could be a problem. All right, so yield. So the 2 locations, this is first generation sandy loam, beautiful ground, 3rd leaf, 4th leaf, 5th leaf, and then the total yield. So what we see is essentially no difference at all in yields. Yeah, I mean, some growers would look at this and go, hey, you're actually getting 400 pounds more where you're not putting it on. But there's so much variability in an orchard when you measure these 10 trees versus that 10 trees. And even though you repeat it 4 or 5 times, you still get, variability. So statistically speaking, these are all the same. So that's in one orchard. Here's the other orchard. This is the replant situation in the real sandy soil. This is where, if it was going to work, to me, this is where you might see something. Again, statistically speaking, they're all the same. If you look at the total yield for all we have so far is 4th, 5th. So this is 6th leaf. We haven't processed everything yet, but here's 300 pounds. Is that real? Statistics say no, it's not real. So essentially, these are all pretty much the same. So I'm curious to see what we get this year in year number 6. So you look at the cost benefit sort of a thing, it's expensive. It's about, $26.50 a ton. So you're looking at pretty high amount of money that you'll be putting out there every year, 10 tons, $265 an acre. So that's a fairly significant expense. So concluding, over 5 years, we spent about $1,300 an acre. We have changed the good things. We've increased cation exchange capacity, increased organic matter, increased soil carbon, increased microbial activity, at least in the top few inches of ground. We see a trend for increasing potassium and nitrogen. And we've also measured not in this orchard, but in different orchard, improved water penetration right at the soil surface. So that's certainly, those are all positive things. Negative is that we've seen an increase in chloride, a decrease in calcium, and I just don't know about the stem water potential or the water status. It's consistent. It's puzzling, but it's consistent. We see a little more water stress where we're putting the compost on, and no difference at all really in tree size or yield after 5 years. So what does this mean? I think that what it means is that this is probably a very, very long process. If it makes a difference, it's going to be a very, very long process. So, we're trying to add organic matter. To me, I guess I question is this really the best way to do it? I mean, are you going to be able to, by spending a couple $100 an acre every year, if it takes 10 or 15, 20 years or something before And I'm just guessing, maybe we won't ever see a difference, but man, that's a lot of money. So I just feel like, I wonder if there's a better way to do it. And I think there is. I mean, I think probably just planting a cover crop, if you really want to build organic matter and grow it that way. I think this newer idea of recycling orchards now, where we're just chopping everything up or chipping everything up and then putting it back in the ground, I think that is probably a real economically feasible way to increase your organic matter, at least for some time. But I still question if we will ever see a benefit of using compost in a good orchard where we just don't really have any problems. And I think that, you know, nowadays growers they're farming almost hydroponically in some ways, where we're drip irrigating every day or every few days. We're putting fertilizer right in that drip hose. They aren't stressing a lot. We're giving them most of what they need. So I don't know. I don't know if you know, increasing the organic matter is going to make that much of a difference. Or again, maybe this just isn't the best way to do it. I don't know. Or maybe it'll be good in some spots. Certainly, if you're organic, you can't use conventional fertilizers. You can get some potassium and some nitrogen. Maybe in these really sandy sites, we'll see something.
What do you make of the water stress, the stem potential? What, as you've thought about that's surprising to me. What do you make?
It is, and you know, sometimes things just show up, and you almost have to blow it off, or just you know, so it's a weird thing that's happening just in that spot. But it's in all three spots, all three locations, different orchards, different growers, different irrigation systems. It's been consistent. And again, I think it might just be that we're adding salts along with that.
The battle from the roots, this osmosis, this pulling water out.
Well, yes, yeah. It's the tree has to, I guess, work a little bit harder to get the water out of the soil and up into the top of the tree.
So, go ahead.
I think that's all the data I was going to show. So,
no. So I mean, what's your recommendation then? I mean, it's a very common, you know, and I've got a little farm. I put compost on when I planted. I mean, it's a very common thing that we do. I mean, so what's your recommendation as people ask you? I mean, doing this work, what are you saying? What do you recommend?
I recommend, well, I don't recommend compost. So if a grower asked me, should I put compost on? My answer would be, we haven't seen benefit from it. Not that it's bad. Maybe there's something to it over time, but I just don't see how it's worth the money.
Okay, fair enough. I've got a question here from Ben, and the question is the noted high chloride in the leaves indicating that the soil EC levels are elevated? Thus, the tree is having to work harder to get water out of the higher EC soil water solution. So along the lines of what we were talking about, what's your thoughts there?
Yes. I haven't measured the EC in the soil, but I'm assuming that's what's happening. Yeah.
Mm-hmm. That would make sense.
There's certainly going to be more chloride and more, and I don't think I showed sodium. It was not a real trend, but I think any kind of these solutes that could be a salt would contribute to that. So and now there's another guy who does compost work for the University of California, and he has a trial in San Joaquin County, and he was saying that he's seeing the opposite. You know, that he's seeing the trees are a little bit less water stressed. But what is interesting was the same guy that does his pressure bombing, the same guy that's been doing my pressure bombing for me, and he was the one that brought it to my attention, saying, hey, you know, this is very consistent. Every time I, you know, do this pressure bombing, that we tend to see, or he's tending to see, that those trees are slightly more water stressed. So I think you know, maybe it's something that could be looked into more. I guess my answer would be, even if it well, I just don't think that adding compost is going to make a big difference in water savings or the water resiliency of these trees.
Yeah. No, I appreciate that, and that it's a lengthy study. How long are you planning on following these blocks?
Well, the growers, he's done. He goes, you answered my question. I don't want to keep doing it anymore. So, it is a pain to spread. It takes a lot of time to spread this stuff and everything. So I could probably convince him to go forward. He was the one who was buying the compost and doing all that. But I think if I were to, find money or somewhere to do that, if I decide to continue on with it, then he would certainly do it. But I don't know. After 5 years, I just, at some point you have to say, if there's an effect, it's pretty marginal.
Yeah. Well, what caught my eye, what I like about it is I think we all of us, any industry people, I mean, Redox, we're a fertilizer company, bionutrient company, whether you're in the compost, gypsum, any industry, we got to measure the dollars we're spending. Are we getting the result? And we just need to constantly do that. That's why it caught my eye, and I wanted to hear more details about it, because I think we just need to do that. It needs to be something that we could consistently do.
Yeah.
But very good. So if you have any more questions on Roger's work, feel free. Justin and I have a few things on our end, how we approach soil health and soil biology and soluble carbon. There's a couple of things we'll review, some research work that we've done on materials in the marketplace on our end. We'll cover that, and then we'll have, if time permits, we'll have a little Q&A at the very end. So feel free to use the chat and the Q&A as we go. So Roger, I'm going to try to, if you will stop share on your end.
I believe I did.
Okay.
So do you want me to sign out?
You're welcome to stay on, and at the end, if there's any questions, it's up to you. We'd love to have you on.
Okay.
All
right. I have an appointment in about 10 minutes, but
okay. Hey, you're okay. You got to scoot. We're good. Yeah, we're fine. We appreciate you being with us today.
All right. I'll listen for a few minutes though. I just and then I'm going to have to bail before very long though.
No problem. So we already went over that. A little bit about Redox. Those of you that are on the webinar might be new to us. Redox exists to create passion and excitement for growing plants. Company based out of Idaho, work throughout the U.S. and internationally in various parts of the world. Today we're just going to cover soil health, a couple principles on it, some agronomics, and then a couple research projects that have been done to measure those effects. So three things. When you think of soil health, it's very, very complicated and diverse and complex subject. I don't pretend to be an expert on it. Justin's not going to pretend to be an expert. We're learning and just trying to learn each day, each week, trying to figure this out of where money, strategy, products should be spent to really get more for our growers at the end of the day. So I'll just cover quickly a couple of facets of this: chemistry, biology, and plant physiology. When we think of soil chemistry, Roger touched a little bit about the importance of that water movement or water penetration. They saw some benefits with compost. That was one thing that they have seen. And I would say if we're trying to improve biology, that would be the very first thing I would look to address, is getting the soil structure in a more balanced, your calcium and magnesium, potassium, depending on your soil type, whether it's sandy loam, a heavy soil, high CEC, low CEC. What can we do, whether it's cover crops, ripping occasionally, down the center, if that's a possibility, or land prep before we plant. That really needs to be the goal if we want to improve our soil health, is improving the drainage. Because when we can improve drainage, we bring oxygen into the root zone, and that is going to drive better biology and nutrient availability. So that would be the first point on that. It's just showing you when we have good flocculation, where calcium is on the clay particles in a balanced way, where we have those particles are held apart from one another, allows air and water to move freely. And what that's going to be is just better root growth. And the more real estate we can get with our roots, the more access to nutrients, water, this interaction between biology and so forth. So what about biology? I like this illustration here because I'll spend just a minute or two on it. Just the picture of the root, you can imagine that's a root hair right there in the soil. And if we have that pore structure, we're potentially in an anaerobic situation where we're not getting the balance of oxygen that not only the root needs, but the biology needs. If we have low levels of carbon or organic matter, that can lead to lower biological levels, and nutrient recycling can be hindered. Nutrient availability in plants, that's a big part of what they need, is those key essential nutrients, and obviously water and that biological interaction. So again, poor structure is going to lead to that reduced biological activity, and that will end up hurting us in the end. As you look at improving organic matter, we just reviewed some of the benefits and not realized benefits of improving there on the compost. Where Redox, what we do is we incorporate forms of soluble carbon, whether that's humic, fulvic, amino acid type products, other carbon compounds with nutrients for a design purpose and to drive nutrient and plant response once they are taken in, whether roots or a foliar material. That's going to encourage soil biodiversity. The slide there, the illustration there on the right, a variety of biology. That's nature's way to be diverse. And when we have proper air-water relationships, an ample supply of different forms of carbon to support the appetites of biology, they feed differently, they have different needs. That can help us in this nutrient release and recycling. I will say on this, before I forget, I wrote a couple notes here to remind me. I was in a presentation. I was a part of a program, I think it was in January, at one of our customers, a grower meeting, where they invited, and I encourage you to look him up. His name is Dr. James White out of Rutgers University. And he specializes in endophytic relationships or microbes, endophytic microbes that are in root, and then have this cycle of in the soil, in the root, out. And what was interesting is, and I encourage you to look at his work, he's been published many times, in this relationship of microbes. The plant will basically farm the microbes at that root hair. The illustration there on the left, it's kind of a picture of that, where they'll push out sugars, they'll push out, proteins, different compounds that will encourage specific microbes. And as those microbes live and grow and they attach themselves to nutrients, the plant, they'll be able to access that root hair, right where it's dividing, and cells dividing, where it's loose and accessible for them to access, pull those microbes in, and then the plant will release ROS, or reactive oxygen species, to break down that microbe, for lack of a better description, is kind of cell wall of it, to access specific nutrients. Those nutrients will be released. The microbe will then, this has been documented, it will exit the root again, and that cycle will start all over again. So pretty fascinating there. But again, it just, that work and how do we capitalize on, in creating an environment where the plant, which knows its needs, how can we just provide it with carbon and provide it with nutrients so that cycle can run as well as possible. So I'd encourage you, it's Dr. James White from Rutgers, pretty fascinating work there that I encourage you to take a look at. And that just goes in with this plant physiology. That's the purple dots that you see. They're just that food, that sugar, those proteins that the plant is pushing out, those carbohydrates, those photosynthates, it is pushing out to feed that microbial population for specific reasons. Some of those secondary metabolites that the microbes are creating are plant stimulating, they're disease suppressing against other fungal or bacterial types. It's just pretty dynamic relationship that we're understanding more and more, but what kind of budget should be allocated, that's where we're figuring that out, across the board. The industry is, and we have some technologies that we like and feel like there's some fits, and we'll share a couple of those. So as we get into a Redox result on one of those products and how we do that, it's a product called H85. It increases microbial diversity. You can see the label on it. It's a 0015. It's a 42% humic acid. It also contains fulvic and long chain. So the purpose of this is to improve microbial diversity, increase that soil health, and it is stable across a broad range of pHs, and it will help with nitrogen efficiency. We've seen that. It's also available in organic form, labeled a little different, a little higher humic acid, a little higher potassium, but it's also available for your organic needs as well. So what's the purpose of this? A little bit is this natural complexing of nutrients that's happening in the root zone that biology is naturally doing, that the plant is feeding this back and forth relationship of releasing these compounds that stimulate biology to chelate or complex nutrients to bring it in. We are reacting those in a product to make that product, as it enters the soil, to create even a stronger environment for biology, as well as nutrient complexing and chelating to pull those into the root hair even freely, and then to utilize that carbon as energy for the plant. So really a process that's already going on naturally in the soil. We're just encouraging it with soluble carbon that you can put on through controlled irrigation, spread down throughout the season, instead of something maybe you can only do in the fall or spring. Is something that can be done season long. So when we're measuring the soil health or the effect of soil microbial activity, you really want to get down to an ROI, or what is the benefit. And this is an example of research that was done in Europe on apple. It was done in Poland, and what's interesting in this is they applied H85 3 times. They did it 3 pounds per acre, total of 9 pounds over the season. And they looked at different biological indicators or markers: dehydrogenase enzyme activity, soil respiration, kind of simple measure there. Looked at different yield parameters, and I'll share just a couple of them. What was interesting in this trial is they looked at a block that they call it a depleted soil that had been farmed apple for many years, compared to new planting virgin soil, and looking at not only yields but also different biological indicators. So you'll see a trend line here in the orange. When they added H85, you see a slight increase in fruit weight there. So we did have an effect on yield, whether it was depleted soil or virgin soil. When you looked at the microbial activity, get my pictures out of the way here. When you look at the microbial activity on that dehydrogenase enzyme, if you look at it closely, you look at the virgin soil on the left and the depleted soil on the right. The orange is with H85 applications, and let's call it a dark red, is without. You do see a difference without H85. You do see it in the virgin soil. Biological activity measured by dehydrogenase enzyme is stronger. The average values are better over the season. And H85 seems to be pretty consistent, whether it's virgin soil or depleted soil, on increasing that enzyme activity. And the next slide here, soil respiration rate, another measurement of biology. Whether it was virgin soil or depleted soil, you saw a nice consistent result with that H85 of increasing soil biology. And you didn't see that without, whether it was, again, virgin soil had at a higher average. That would make sense, less farm soil, less cultivation, less oxidation, probably more organic matter available for that less disturbed area for that biology to thrive. I'm going to pass the mic to Justin. Let me unshare. Let me see here. One second, Justin. This thing locked up on me here. Stop share.
Well, hey, good morning, you guys. My name is Justin Rose, and I'm an agronomist here with Redox, based out of Fresno. So let's get into it. What this Redox result is a root development with 2 of our products. One is Root RX, and the other is Root Tex. All right, so who we did this with? We did this with Wes Asai. He's out of Stanislaus. He's a former University of California advisor. And what we did it on was young potted nonpareil trees. They were planted the 6th of February, 2019, and then they were harvested on the 13th of June, or of July. So about 5 months during that growing season. The soil is prime soil, perfect. And then, again, we started in the spring, we ended in the summer. So here's a Rootex. Let's go over that. Rootex is a 6-46-6. So it's our high phosphorus fertilizer. It contains soluble carbon, and then it also has 8 key amino acids, 6 of which help particularly in lateral root growth and root branching. So let's see what RootRx has to do. RootRx, it's a 005. RootRx is botanical extract from willow and black walnut. And what those compounds do, stimulates root growth, the root metabolism, and then the overall quality and quantity of those roots. Let's see how we got into it. So what the study was, we did 2 different applications during the 5-month period, and we wanted to evaluate 3 different parameters: the top growth dry weight, the root growth dry weight, and then the trunk caliper. So as you can see here, the different applications, there's 4 types. We did the grower standard of a 20/20 twice. Then we did Rootex with the grower standard, 2 pounds and 1 pound as a standalone. Then we did RootRx with the grower standard, 1/2 gallon and 1/4 gallon. And then we did a combination of the 2, Rootex, RootRx, and the grower standard. Here's some cool images. Wes does a great job. He really gets into the details of it. He likes to record what he does, to show guys like us and all the farmers, he does do the extra work and takes an extra step while doing research. Here, he's mixing the soil, getting them right, getting them planted. Here, something that's unique when you put them in these pots, they're 15-gallon pots, right? And he put foil all the way around each pot. Reason being is to reflect the sun so he doesn't get a bias in the roots. So, any type of harm that sun may do to the black, heat up or something may damage the roots. The foil helps to prevent that. So I thought that was pretty cool. That's something I'd never seen before. Then right here, so he took them out of those pots, he cleaned them thoroughly. You see the root weight, the dry weight. And then he, measured the trunks, and then he put the roots in an oven, to make sure they were real dry. So he does the extra steps here. All right, so this is the information we want to see. How did these products, how they do out in the field? So this shows the top dry weight. There's about 0.10 pound difference in the grower standard to the Rootex, and then you see a little drop from the Rootex to the RootRx. But what you see, the cool thing here is the Rootex and the RootRx, they combine to do about the same as that Rootex. That's kind of interesting. The dry weight. This one I think is really interesting because it shows grower standard 0.38 pounds. And then the Rootex, it shows an increase of 7.9%. Then the RootRx, we get a 26% increase as a standalone. And then combined with the Rootex and the RootRx, we get about the same. So, I wonder what's going on here. This next slide kind of puts everything together. I like this because it shows the trunk caliper. And what it's showing is a 14% increase over the grower standard with Rootex and RootRx. And again, those 2 products, they're helping create a better root zone by creating more feeder roots to mine the minerals there, therefore giving off, like Jared said, they're giving off different compounds that are attracting those microbials to help break down nutrients. So, and to take just a step back, let's look at that 26.3% increase. You guys, this is about MSRP, a $75 increase per acre in spend, adding this combination of Rootex and RootRx. But that 75% or that $75 is resulting in a 25%, give or take, increase in root dry weight. That's pretty huge. So that being said, the reason we wanted to show that is to hit on those points. Creating stronger, better quality roots is going to help mine the soil. Therefore, that's going to create a better zone in that root zone, for the growing season and for the future. So here's another one on a product Jared got into already, which is H85, that's done in strawberries over in Oxnard. Yeah, and this is all organic also. So what we did here, it was H850015. So you got a little bit of potassium in there, but this is what we did. We did a grower standard, which was, a combination of NPK organic pellets, about $300 per acre. We did a grower standard of H85, 5 pounds per acre applied every 14 days. So that resulted to, we applied it from August to November. So we put about 40 pounds of H85, during that 4-month period. And then the mulch application. The mulch was a combination of the rice hulls and some other stuff put together. And that was 3,000 an acre. So let's see what we looked at here. The trays per acre. Grower standard plus H85, it resulted in quite a bit increase. The mulch stayed right at 31, right under 31. Grower standard went above that. So that being said, we're 17% over the grower standard and 18% over mulch. What does that look like, to the grower? The grower noticed a profit of 10 times what he did to the grower standard. That being said, that results from better root environment. Those roots are able to grow. There's more nutrients available for those feeder roots. So that's what we really like to see there. Now I'm gonna kick it back over to Jared here. Click in, Jared.
Okay, thank you, Justin. Okay, a couple more slides to finish it off. I like about that strawberry example, if I can get down to that, what I like about that is it's about nutrient availability, obviously encouraging healthy biological conditions in an organic cropping system. Maybe we don't have the most available forms of nutrition that can be put on in a standard organic practice. So anything we can do to increase nutrient availability consistently through the season and multiple applications, I think is a good strategy. And at something like strawberries, that the price per tray is substantial. So any yield increases showed a pretty dramatic effect in the bottom line for the grower. So that's pretty interesting. I think just to finish it off, move these pictures out of the way. I think just to recap, look, it's the Redox technology. H85 is one. There's a few other materials. F70 is a high fulvic acid material that we also carry. A new product you look for, expect. It helps with phosphorus and nutrient availability. It's a high amino acid carbon product. We are releasing more materials in this space of improving biological and soluble carbon in the root zone. H85 is obviously our flagship, but we included Rootex and Rooterex, as Justin mentioned, because they do contain carbon, and they drive that healthy root zone that's going to improve your soil biological conditions there in the root zone, the rhizosphere. I just think lastly there is just to capitalize on, look, it's not one approach. It's not putting on one magic product that's going to make something like this happen, good results. It's getting the water right, getting the soil structure right. That takes planning, takes execution, and adding key inputs. It can make a difference. And we are big on getting the research, showing, presenting things to the market that we know are consistent across crops. So really just an eye on keeping the soil diverse, making sure oxygen and water is moving consistently through that profile. And again, anything we can do to increase root real estate we have seen in the research and just on the farm, working with customers, consultants, customers, that's where ROI can be achieved. So one last product I failed to mention, I think I've got it right here. This is a new one for us. It's pretty interesting. It's called Dermaplex Plus in this space of soil health, soil biology. It does have H85 in it that we talked about. There's some of that in this product, but really what sets this product apart, it's a trichoderma metabolite extract product. So trichoderma, beneficial fungal, there's very common in soil. There's lots of different strains of it. Some are more plant responsive and plant stimulating than others. But really what's in these metabolites? They contain key enzymes, they contain plant stimulating compounds, auxin-like stimulation. They can help the plant deal with different pest pressures, things like that. That's what trichoderma naturally do in the soil. What we've done is taken a strain that has been most stimulating to plant growth and yield response, cultivate that, and then extract those metabolites and include that in this product as a concentrate. So the rates on this product, you're looking anywhere from, it depends on your crop. If we're talking nuts, we're talking almonds. We've done several quart applications this year here in the South Valley with very good response. But you could even do it lower than that, say 16 ounces, 20 ounces, early season, follow that up with another, and you should see some nice response there and beneficial biological activity. So anyway, that's the last new product there in the arena that Redox brings. It's a trichoderma extract product as far as capturing those metabolites and incorporating them into the soil. Okay, so any questions? That was the last that we have, and I'm looking at the time. We're at 12:02, so we did pretty well there. I'd like to thank Roger Duncan from Stanislaus Farm Advisor for his contributions today. Appreciate him. This webinar will be on the YouTube Bionutrients, Redox Bionutrients page. And once we get our CCA, CE units approved and sent to us, we'll forward those on. Everybody who has registered and participated today, you'll be getting that. Any final questions or thoughts anybody might have? Let me check, see if we have anything unanswered. Justin, can you help me on that? Okay, well, very good. We'll let you get to your day. Thank you for participating, and we'll talk to you down the road. Thank you.
Thank you, guys.